Hi, everyone. Welcome again to today's episode of Let's Talk. I am excited to host our guest for this episode, Jessanne Lichtenberg. In our previous video, she shared with us her journey to pursuing a career in biomedical engineering. She also shared about some of the achievements, challenges, and the things that has inspired her so far. Definitely check that video. You'll see the link to the video in the bio. On that note, welcome again, Jessanne.
Thank you for having me.
So to start with, would you like to tell us briefly about what your research is about,
Sure.
what you do for your research?
Yes. So basically, I am working on developing a breast tumor on a chip, which is a 3D microphysiological model that is used to answer kind of basic cancer biology questions. So my project is, focused on studying how breast tumor cells can collectively invade and migrate into the extracellular matrix, which is the initial step in the metastatic cascade, where, metastatic breast cancer is kind of the most fatal form of breast cancer, right? So that's where the survival rate is the lowest, unfortunately. And so, a lot of breast cancer research previously has focused on single cell migration. But we know now that most breast cancer migrates in, these collective units where the cells are all coordinated with each other and they're communicating with each other when they're migrating. And so, this makes it a lot more complex to study. And, in these collective units, there's a specific type of cell called a leader cell, and they lead the other cells to migrate in a particular direction. And so I'm looking at how different mechanical cues in the extracellular matrix promote these leader cells to initiate migration.
Wow, that is great. And you mentioned something about a microphysiological model. Could you explain more about that and how you apply the model to your research?
Yes. So when I say a microphysiological model, so what it is a 3D dynamic platform that we use to study like cell to cell and cell matrix interactions in the lab. We try to mimic the features of the in vivo conditions as close as possible. And so these are on the microscopic level. So like the height of the like micro channels are about 100 microns, which is about the width of like a human hair or a sheet of paper. So it's very thin, you can only see it under a microscope. But allows for like the very fine resolution that you cannot get from like using like animal models, for example. And so these are, clear devices, so that makes them very compatible with different microscopy techniques. And it's an advantage to 2D cell culture models because, normal 2D assays, you have cells like on like tissue culture plastic, right? A very hard surface that is not at all like how cells are in the human body, right? They're in a soft kind of squishy matrix. They have different fluids going on around them. And so our models allow us to kind of mimic those important features.
Wow, that's great. So you also talked about the metastasis, and can you explain more about how that process occurs?
Yes, that's a great question. So when we think of like metastatic breast cancer, it's like stage 4 breast cancer, right? Okay. So at that point, these cells have left the primary tumor and they are now in a secondary site, which can be like the lungs, the lymph nodes, the bone marrow. And so when metastasis occurs, so the tumor cells at the primary site, so if that's breast cancer, then we're talking about like the mammary gland tissue. So they will leave, the primary tumor site, and then they will travel into circulation. So either like cardiovascular system or maybe like the lymphatic system. And they will use that, as kind of like a highway to go to somewhere else in the body. And then they will form the new tumors at the secondary sites. And so, when cancer metastasizes, it makes it very hard to treat, because treatments are most effective when it's localized, and you have more control over, what the cancer cells are doing, where they, are, how big they're growing. But once it starts to travel into different areas of the body, it becomes so hard to treat. And so the goal of my project, is that, I hope that the outcomes will aid in the development of therapies to prevent metastasis. Because if we can, prevent the initial step, which is, the invasion into the matrix, collectively, then we can prevent the whole cascade from occurring.
Oh, okay. Wow. That's great. So how does the aligned collagen fibers promote migration?
Yes. So I mentioned like the extracellular matrix and collagen fibers. So in the breast tissue, you have cells in the extracellular matrix. So you kind of think about it like my favorite analogy that I heard, it's like a bowl of spaghetti. So if you imagine the noodles are the extracellular matrix, and the meatballs are the cells, and the like pasta sauce is the interstitial fluid coating everything, right?
I'm in the process of imagination, so
yes. And so that is kind of how your cells are, in the extracellular matrix, right? So they are in contact with these fibers, like in breast cancer, it is normally collagen 1 is the main kind of ECM fiber that we see. And so this is like a scaffold that these cells, use. It gives structural support, and it's also how the cells migrate. So they, literally like grab onto the ECM fibers and move. And so, studies have shown that in areas with a lot of tumor mass, you start to see more aligned collagen fibers. And that literally creates like pathways for the cells to be able to migrate more efficiently. It's kind of like giving them literally a road to travel on, instead of just like say like an open field, where they could go anywhere, but then they have these like aligned fibers that are like, oh, go right here, and so that really aids in the metastatic cascade when you have these aligned fibers.
Wow. That's great. So how does the research in general improve the knowledge on cancer? Cancer is, I think, one of the leading deadly diseases these days. And I'm glad that you and other researchers are studying more about these things and how we can be able to control them. So how does the research you do also contribute to the knowledge on cancer and all of that?
Yeah, so my research, I'm hoping that it will give us more insight about leader cells, because this is kind of new in the field of cancer. We really don't know a lot about their phenotype, how they work, how they develop, because it is a unique subset of cells from the other cancer cells. And so understanding, more about the leader cells will help us to identify what is initiating metastasis. And like I said, that's then the key to developing therapies to prevent metastasis from occurring. And then also, on the engineering side, my project, like I said, is using these 3D microphysiological models. And so I'm working on developing, a new microfluidic model for my project specifically. And so, I hope that other people can, use this model that I've created and answer other questions about cancer biology, even when I'm done with my PhD.
Wow, that is awesome. So you mentioned when you are done with your PhD, what are some of the plans you have in the future, if you don't mind sharing it with us?
Yeah, I'm not 100% sure,
I know.
but I'm pretty sure that I want to continue a career in
Okay.
academia, because I really like being in a university setting.
Mhm.
I really like working with undergraduate students and helping them figure out their career and learn new skills in the lab. And so probably a professor of some sort one day.
Go for it. Go for it. Mhm.
Yeah, so we can both be professors.
I didn't see that coming, but oh, great. Thank you for coming on the show, Jessanne. And again, we wish you the utmost best in your career. And we know that you will do awesome. We need more people like you to be in academia, to mentor the young ones that are coming and do the wonderful and cool research you do to improve the knowledge that we have on cancer. So yeah, thank you. And thank you, viewers, for staying with us to the end of this episode. Stay tuned for more videos with different people in academia, both graduate students, postdocs, and also with faculty members to talk about some of the issues in academia and how they have been able to overcome some of the issues that aren't talked about most often, and also to share about all the research that are going on around the world. Until our next video, let's enjoy science together. And I remain your host, Emelia. Thank you.